Hello students, welcome back to my channel. Today I am starting with a new video, and this video is basically on the experiments on photosynthesis. So in this video I will be discussing about various experiments which are there in Icse class 10 syllabus, and all these experiments are from photosynthesis. So without wasting time, let's get started with the video. Now before starting with the experiments, first you need to know how to remove the starch from leaves or from a plant. That process is known as destarching. The word de-starching, de means to remove, and starch, you all know that starch is produced as a byproduct of photosynthesis, just like the glucose. Okay, if you haven't watched the videos on my channel on photosynthesis, then please go and watch those videos first and then come back to this video for better understanding. So as I was telling, that destarching means to remove the starch from a plant or from the leaves. Okay, now how to destarch? First you need to take a plant. Okay, and what you need to do is you have to take this plant, and this you should keep this plant in dark condition. In dark, that means without the presence of sunlight. In the absence of sunlight, you need to keep this plant in dark for 24 to 48 hours. Okay, and with this process, the leaves or the plants will become totally destarched. That means the starch which is present on the plant or the leaves, it will be totally removed. Okay, during this whole period, all the starch from the leaves will be removed and to the storage organs, and the leaves will not show the presence of any starch. So a destarched plant is one whose leaves are totally free from starch, and the plant is destarched by placing it in the dark for 24 to 48 hours. Okay, I think this is clear to you all, because before starting with the experiment, how to destarch a plant, this is very important. This concept, it should be crystal clear to you all. Okay, now next concept is how to test a leaf for starch. That means how will we test whether in a leaf the starch is present or not. For this, the test is iodine test. Okay, the test that we perform to see whether the leaf or the plant contain any starch or not, we perform iodine test. First, what we need to do is, first we need to dip the leaf in boiling water. Okay, for a minute we need to dip the leaf in a boiling water. And why we are performing these steps? We are performing these steps so as to kill the cells of the leaves. Okay, this is the first step. So as to kill the cells of the leaves, we are dipping the leaves in boiling water. Clear? This is step number 1. Then next, what we are doing in step number 2, we are boiling the leaf in methylated spirit. Methylated spirit, or you can use ethanol, any one. Okay, either you can use methylated spirit or ethanol. This is step number 2, and we have to keep that test tube containing ethanol or methylated spirit and the leaf. We have to keep that test tube in water bath. That means in a beaker containing water, just similar to this picture. Okay, and what will happen in this step? After keeping the test tube in the water bath, what will happen is the chlorophyll which will be present in the leaf will totally be removed. And due to the removal of the chlorophyll, now the leaf will become hard and brittle. That means it will be broken easily. Then next we have to place that leaf again in the hot water just to soften the leaf. And finally, in step number 4, we have to take a dish, and in that dish we have to pour the iodine solution, and in that iodine solution we need to spread the leaf. This is basically the 4 steps. I am again repeating the 4 steps. In the first step, you need to take the leaf which you want to test whether the starch is present or not. That leaf you need to take and you have to boil that leaf in the water for a minute. Okay. And why we are performing this step? Just to kill the cells of the leaf. Then in the next step, what we are doing, we are taking that same leaf and we are putting that leaf in a test tube containing methylated spirit or ethanol, anything. Okay. And then we are putting that whole test tube containing the ethanol or methylated spirit and the leaf into a water bath. That means a beaker containing water. And by this process or by this step, we are actually removing the chlorophyll that is present in the leaf. After the removal of the chlorophyll, what will happen? The leaf will become very hard and brittle. It will be easily broken down. In the third step, what we are doing? We are placing that leaf again in the boiling water just to soften it. And in the final step, the fourth step, we are taking a dish containing iodine solution and then we are pouring that leaf or spreading that leaf in that iodine solution. Now what will be the result? Now if we see that blue black coloration, if we see that there is a bluish black coloration, okay, then we can conclude that starch is present. So if starch is present, then this test will indicate a blue black coloration. And if we see that there is a brown coloration, that means starch is absent. So basically, if we see that there is a bluish or bluish black coloration, then starch is present. And if we see brown coloration, then starch is absent. So this is basically how we test a leaf for starch with the help of iodine test. So if in exam, if it's asked that how can we test a leaf for starch or explain the process or procedure of iodine test, you need to write all the steps and the observation of the result. So you need to understand this particular slide very clearly. Okay. Now come to the first experiment. The first experiment is to show that chlorophyll is very much necessary for photosynthesis. Without chlorophyll, photosynthesis is not possible. Okay. For this, what we need to do is, see, there are various steps. First, take a plant with variegated leaves. That means a plant where the leaves, some leaves have green and non-green areas. That means a leaf will contain some green portion and some non-green areas. Okay, that type of plant is known as, or that type of leaves are known as variegated leaves. We need to take a plant of containing that type of leaves. For example, croton. Croton is a plant which has variegated leaves. That means leaves have some green portion and some non-green portion. Then in the second step, we need to discharge the leaves by keeping the plant in a dark room for few days. For 1 or 2 days, I have already told you how to discharge a plant or leaf. Then after discharging, we need to place that plant in the sun. And after few hours, we will pluck one leaf from that plant. And we will make an outline on the paper and mark green and non-green areas on the outline. And then we will test the leaf for starch. See this picture. Here, this is the green portion, right? This portion is green. This is a green portion and this is the white portion. Okay. Now, if we perform iodine test in this leaf, with this leaf, then the result will be this one. We will see that only green part turns bluish in color, blue in color. Okay. And the white portion has turned brown in color. And what we have seen in the iodine test, those portion which will show bluish black or blue color, it will show the presence of starch. The green portion which contains chlorophyll shows that there is presence of starch, and the brownish coloration which was white in color, that means the absence of chlorophyll, there no starch is present. That means no photosynthesis has taken place. Why? Because we know that the end product of photosynthesis is starch. Glucose is a type of starch, right? So No photosynthesis has occurred in white portion, which shows the brown coloration after iodine test. Because there is no presence of starch, that means no photosynthesis. Only the green portion, which shows the presence of starch, that means there the chlorophyll is present and photosynthesis has occurred. So with this experiment we can conclude that chlorophyll is very much necessary for photosynthesis, because the portion where chlorophyll is present, which is green in color, only in that portion we have found starch is present with the help of iodine test. Clear? Now comes to the second experiment, which shows that sunlight is very much necessary for photosynthesis. How? See, first we need to take a plant and again we need to destarch the leaves by placing it in the dark for 1 or 2 days, okay? Then we have to cover one of its leaves with black paper on which a design is cut. See, a design like a star is cut in that black paper and we need to cover the leaf. Okay, this is the original leaf. We are covering that leaf with a black paper where a design is cut. Suppose the design in this case is a star. Then we need to place this plant under the sun, and after few hours we will test the leaf which is covered by the black paper for the presence of starch. That means simply we will test this leaf iodine test. Okay, we will do iodine test with this leaf so as to see the presence of starch. Now in the result we will see only the parts of the leaf which could get the light through the cart, okay, through the cart design, as well as those portions which are left uncovered by the paper has turned bluish black. See, only this portion which has got sunlight through the cutout, this portion, then this portion, that means greenish portion which has got sunlight, that portion shows bluish black in color. That means the presence of starch. And the portion which has been, the black portion which was covered, okay, which didn't get any sunlight, this portion show brown coloration. That means what? Absence of starch, right? That means there in the brown portion where it was covered, or the portion which was covered by the black paper, there photosynthesis has not occurred. That means sunlight, where the sunlight is not present, there the photosynthesis is not occurring. That's why it is brownish in color, which shows that there is absence of starch. And the portion where sunlight has reached, the green portion, the photosynthesis occurred and the starch is present. So from this experiment we can conclude that sunlight is very much necessary for photosynthesis. Clear? Now the next experiment, which shows that carbon dioxide is necessary for photosynthesis. For this what we are doing, first we are taking a plant and again we are destarching the leaves in the same way. Then we are inserting a part of a leaf, okay? Suppose the half of the leaf, we are inserting that through a split cork into a conical flask containing potassium hydroxide, okay? See the picture. This is the picture. This A portion is the picture. This is a plant. See, this is the plant, and we are actually placing a part of the leaf, just this. This is the magnified view. We are placing a part of the leaf, like half of the leaf, inside that conical flask which contains potassium hydroxide. Okay, potassium hydroxide is a base. And then we are leaving this plant in sunlight, and after few hours we will again perform the iodine test so as to see whether starch is present or not. We will find the result as the part of the leaf which was exposed to the atmospheric air, that means only this portion, the outward portion, okay? Only those portion which has been exposed to the atmospheric air, that portion will become bluish black. That means it shows the presence of starch. And the portion which is inside the conical flask, okay, here, it will become brown in color. Okay, it does not become blue black. Why? Due to the presence of potassium hydroxide. This experiment is very, 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 very important from Icse point of view. Often this experiment comes in board exam. Why? What is the role of potassium hydroxide? Why the portion of the leaf which is present inside the conical flask didn't become bluish black? Why? Because potassium hydroxide actually absorbs carbon dioxide. This potassium hydroxide absorbs carbon dioxide. That's why inside the conical flask, as potassium oxide is present, so this potassium hydroxide has absorbed all the carbon dioxide available inside the conical flask. That's why photosynthesis cannot take place inside the conical flask, and so the portion of the leaf which is present inside the conical flask doesn't become bluish black. That means there starch is not formed, or there is absence of starch due to the absence of photosynthesis, because photosynthesis didn't occur, right? Due to the absence of carbon dioxide, as potassium hydroxide has already absorbed the carbon dioxide which is present in the conical flask. That's why starch has not formed, and as a result bluish black is not seen, because due to the absence of starch. Clear? So in this experiment we have found that since CO2 was not present in the conical flask, photosynthesis couldn't occur. So from this experiment we can say carbon dioxide is very much necessary for photosynthesis. And the last and the fourth experiment is to show that oxygen is produced during photosynthesis. In my previous video, where I have discussed about photosynthesis in detail, I have already told you that oxygen is produced along with the glucose or the starch or the food material. Oxygen is produced during photosynthesis. So see the experiment. We will take a plant, water plant, okay? For example, hydrilla is a water plant. This is the picture of hydrilla. So we will take some water plants like hydrilla in a beaker. Okay, this is the beaker. And this beaker contains pond water. And we will cover, or we will place this hydrilla in a funnel, short stem funnel. This structure is funnel, okay? We will place hydrilla inside that funnel. We will cover hydrilla, okay? Now we will insert a test tube, okay, on that funnel, which also contain water. It will initially, the test tube will totally contain water, pond water, okay? And this is the afterward result. And we need to ensure that the level of the water in the beaker is above the level of the stem of the inverted funnel. That means the water level should be above this funnel, okay? This is a precautionary method. Often question comes like, state one precaution during this experiment. So this is that precaution, that we need to ensure that the level of the water in the beaker is above the level of the stem of that funnel, okay? Then we need to place that apparatus in the sun for few hours, and after that we will see here the bubbles are coming. I think you can see that the bubbles are coming, right? Bubbles of the gas will collect in the test tube, the above. And if we test that gas in the test tube, we will see that if we take a glowing splinter into this gas, then it will burst into flame. And we know that oxygen actually supports combustion. As oxygen supports combustion and as the glowing splinter bursts into flame, so we can say that this gas is nothing but oxygen. So in this experiment, we are seeing that we are taking a water plant, hydrilla, and we provided pond water, we are providing sunlight, everything in the plant, chlorophyll is present, so everything we are providing. So for this bubble actually proves that photosynthesis is occurring, and due to this process of photosynthesis, a gas is produced, okay, at the top of the inverted test tube. And when we test that gas with a glowing splinter, we see that it bursts into flame. And as oxygen supports combustion, so we can say that the gas which is produced is oxygen. So from here, from this experiment, we can say that during the process of photosynthesis, oxygen is produced. Clear? So I think all these four experiments are crystal clear to you all, because these four experiments are very, very, very important from Icse or any board exam point of view. So this concept need to be clear very, very, very clearly, okay? So I think this topic overall is very clear. I have completed the whole topic of photosynthesis. I think you have liked the videos of my channel. If you think that my videos are helpful and they are informative, then please like and subscribe to my channel. And please stay tuned to my channel for more such videos. I will be back with a new video very soon. Till then, thank you and bye-bye. And thank you for watching the video.